Hello everyone, this is Rahan Chanfir. Today I am presenting my paper, which is stage-specific co-expression network analysis for cancer biomarker discovery. So, whenever there is any cancer research to determine the cancer biomarkers, molecular subtypes, prognostic signatures, or therapeutic treatment, the most commonly used dataset is the gene expression data. In the gene expression data, gene expression means the quantification of the transcript product which is produced by gene for that particular sample or a particular cell. And if a group of genes have similar type of expression among multiple samples, those genes may form pathways, complexes, or regulatory signaling circuits. So cancer development is divided into 4 stages based on how the tumor is developing. So in stage 1, the cancer tumor is relatively small in size and it is localized to an organ. In stage 2, it is a bit advanced. The tumor size is a bit advanced, but it's still localized. In stage 3, the tumor is grown to an extent that it has reached the lymph nodes, and in stage 4, it has already reached to other organs, cancer cells, which means that the cancer is metastasized. So we are using co-expression network. So what is co-expression network? In this kind of network, the nodes are the genes and the ages means that the strength of the correlation between genes. So if two genes have an age in the co-expression network, that means that those two genes are highly co-expressed. They have similar co-expression profiles in all the samples that have been used. And there are 2 steps to construct a co-expression network. The first is apply a correlation method, and the second is to apply a threshold. So in this example of 3 samples and 10 genes, at first Pearson correlation is applied, and after that a threshold of 0.8 is applied to construct the adjacency matrix. From there we have our co-expression network. So we are targeting here 2 hypotheses. The hypothesis 1 is the co-expression network of multiple stages and the subnetworks that we are extracting from those co-expression networks. If a module is conserved in co-expression networks of all other cancer stages but not in normal samples, that means those cancer modules might be a trajectory specific biomarker. They are related to cancer development or cancer progression from initiation to stage 1 to stage 2 and etc. and to metastasis. And those cancer modules which are not conserved in any other cancer stages, that can be considered as a cancer biomarker only related to that particular stage, and thereby they can be called as stage-specific biomarkers. So this is what our overall process flow looks like. So we are dividing our gene expression data into 5 different groups based on stage information and normal tissue samples. Then we are constructing a co-expression network using correlation method and thresholds. And after that, we are extracting the modules from the co-expression network of 4 stages using module detection method. And we are using the modules from 4 stages and clinical data to and we are applying survival analysis to find out which of those modules are prognostically significant, and then we are applying module preservation analysis to see that whether the modules are coming from one stage but also they are preserved in different stages or also in the normal gene co-expression network or not, to see that whether they can be called as stage specific or trajectory specific biomarkers. So in here I want to explain some related works. In the work by Palaniapal et al., what they did was they constructed a stage-specific protein-protein interaction network using some driver genes, and from those they applied network analysis to find out some hub genes which are then said to have relation to progression of colorectal cancer. In the work by Lee et al., they constructed stage-specific co-expression network of 4 cancers, and then they applied Jacquard similarity and rank analysis to see the network topology changes among different stages of those co-expression networks. In the work by Pradhan, what they did was they constructed a stage-specific PPI using differentially expressed genes and differentially methylated genes, and then they applied network analysis to find out hub genes, which are then said to be essential for prognostic target for lung cancer. In the work by Lalremia, what they did was they constructed a stage-specific co-expression network using some query genes, and they listed out the highly ranked genes which are highly co-expressed with the query genes which are called as highly ranked genes, and those were later used in network analysis, differential gene expression analysis, and SVM, which resulted in 17 biomarker genes for ovarian cancer. So what makes our work different from theirs is that we are using normal tissue samples gene expression data into our analysis, and we are also using a module preservation analysis to see whether the modules coming from one stage is preserved in different stages or not. So for the data collection, we collected TCGA breast cancer data from Xena browser, which contained 32 cancer types, TCGA cancer types, and multi-omics data. And we are using gene expression data, which is RNA-seq log2 RSEM normalized count. And in the clinical data, there were multiple attributes for each of the patients, and we are only using overall survival and pathologic stage. And in the data pre-processing phase, what we did was we removed those samples which did not have any stage information, and so these are the these are the sample distribution of different stages and normal tissue samples. And for removing the genes, we removed only those genes that contained low expression values among most of the samples, which means that the criteria that we used was we looked at only those genes which had normalized count less than 10 for 90% of the samples, and we got different number of genes from each of the different group of samples. We took the common from them, and then we removed these many genes from the original number of genes, and so the remaining genes are around 16,000, which are later used in our network construction phase. So in our network construction phase, we used WGCNA, which is weighted gene co-expression network analysis, and in there we used soft threshold to construct the co-expression network. So which means that we applied a Pearson correlation on the gene expression data, and we are applying a exponent factor on the absolute value of Pearson correlation to generate our adjacency matrix, which is later used as co-expression network. So when we are using soft thresholding to generate our co-expression network, there's an issue of how to choose the soft thresholding exponent, the beta value, and it is chosen in such a way that the resulting network would have maintained a scale-free topology, that a network would be a scale-free network, which means that the probability distribution of the connectivity of the network would approximately follow a power law. And if we apply logarithm on both sides, we have a approximately linear equation, which is then being trying to fit using a linear regression. And from the linear regression, we have a model fitting index R square, which would denote how close it is to the regression line. In the right side, we have two examples using beta value of 7 and 6, and as it can be seen, the only beta value 6, the degree distribution would be closely related to the regression line, resulting in a higher value of R square. So 6 is the appropriate value for network construction using soft thresholding method. So in our case, we used a threshold for R square, which is 0.9, and we varied our value of beta from 1 to 30, and we chose only that value of beta that was the first one to cross our threshold, which is 7 here. We did not choose the rest of the values here because that would result in a much lower connectivity, as it can be seen from the second figure here. So for each of the groups of samples, we found a different number of soft thresholding exponent here. And after constructing our co-expression network, we then move to our module detection phase. And in here, we use the adjacency matrix of the co-expression network, and from that is used to construct the topological overlap matrix based similarity matrix. And from the Tom based similarity matrix, dissimilarity matrix is then calculated using by subtracting 1 from it. The Tom based similarity matrix tries to calculate the similarity between nodes by using the common edges and the edge weights between them. And Tom-based disseminated matrix works as a distance matrix that can be used in the clustering algorithm, and here hierarchical clustering algorithm is applied, which results in a dendrogram, and an appropriate tree height is found using a dynamic tree count algorithm, and that tree appropriate height is then applied to cut the dendrogram, which would result in different trees, and then they were grouped into different modules. Now that we have our modules, we want to see that whether they are prognostically capable. They are able to prognostically differentiate patients, and to do that, what we used is we used those module genes as variables to divide that whole patient groups into two groups, group A and group B, using k-means clustering with k equals to 2. And now that we have two groups, we then apply log-rank test and Kaplan-Meier test to see that whether they are prognostically significant. And the criteria that we used here was the log-rank p-value either less than equals 0.05 or hazard ratio greater than equal to 1. So in the example Kaplan-Meier test here, we can see that the group A and group B are clearly distinguishable from one another with very low p-value and comparatively high hazard ratio. So in the module preservation analysis phase, we used netrep, which is a R package that is compatible with WGCNA. We used this one to do our module preservation analysis test. It uses the 7 equations which are labeled as MPI statistics. The input that it takes is a module genes and the gene expression matrix, correlation matrix, and adjacency matrix of two networks. One is the discovery network, from where the module is gotten from, and the second is the test network, in where the module is being tested on. And the output is, it gives out 7 different p-values based on the empirical null distribution using permutation test. And the criteria that was used is, if all of the p-values coming from the 7 MPS statistics happens to have less than 0.05, then it is decided that the module is also preserved in the test network. So let's look at the results. We found 7 different beta values for different group of samples, and we also found different group of modules in the module detection phase. For example, stage 1 contributed to 293 modules, stage 2 contributed to 267, and so on. We also look at the highest number of genes a module can have. So stage 1 module, the largest module contained around 1300 genes, and the smallest module was rather determined in parameters. So we believe the 4 was the smallest a module can be. And in the right side, this is coming from the WGCNA from index 0 to 293, and each of the modules have different number of genes in them, and the cluster dendrogram and the module assignment using colors. This is coming from WGCNA. So in the survival analysis, we have different number of modules that happen to be prognostically significant. So for example, in the stage 1 modules, from 293 modules coming from stage 1, only 26 of them happens to be prognostically significant. They passed the survival analysis test. And also, as you can see, stage 4 contributed to a lot more modules than the rest of them, so they are also contributing to much higher prognostically significant modules. And the right side, this Kaplan-Meier curve is coming from stage 1 for a particular module, which is module index 103, and module index 103 was able to distinguish cancer patients with a very low p-value and very comparatively higher hazard rate. So module index 103 from stage 1 can be considered as a module with a prognostically significant capability. Now let's look at the MPS static module preservation analysis results. So in the right side, we have an example. So module 1 from 10 is coming from stage 1 and are being tested on stage 2, and it can be seen that only module 5 has the p-values for all the 7 MPS statistics that had p-values less than 0.05 for all the 7 MPS statistics, which means that only module 5 is preserved in stage 2 among 10 modules. And the left side contains the summary of all the preservation analysis. So for example, among 293 modules, 74 of them happen to be preserved in stage 2, stage 3, stage 4, but not preserved in normals. And 30 modules out of 293 were preserved, not preserved in any of the other stages. So And among 74 modules, 7 of them happen to have prognostic capabilities, and from the 30 modules, only 1 of them happen to have prognostic capabilities. So which means that the 7 modules coming from stage 1 are potential trajectory specific biomarkers, and 1 module coming from 30 is our potential stage specific biomarkers, and the rest is the same goes on for stage 2, stage 3, and stage 4. So in conclusion, we are proposing a computational pipeline to discover biomarkers. And the kind of biomarkers we are finding are 2 kinds of biomarkers. One is the stage-specific biomarkers, which are related to that particular stage, either stage 1, stage 2, stage 3, or stage 4. And the second kind of biomarker is trajectory-specific biomarker, which means that the biomarker is present in all the 4 cancer stages, but also not in the normal patients. And we found a total of 25 trajectory-specific biomarkers and 83 stage-specific biomarkers. And the future work ideas is that we would like to use multiomics data and to generate a heterogeneous network to generate the modules. And we would also like to validate our trajectory-specific biomarkers using pseudo-temporal orderings or temporal graph analysis. And we would also like to apply graph-based ML to find our modules in a different way. And this work, and I would like to thank my advisor, Dr. Ananda Mandal, and also would like to thank MLDAG members, Abdullah Mahmoud and Master Silvan. I would also like to thank BRCA members, and this work was supported by NSF Career Grant. Thank you, everyone.